Good afternoon, everybody. I'm aware that I'm the last speaker between yourselves and drinks, so I'll stick to my 15 minutes. I'm here to present Valo Therapeutics, an immuno-oncology company that's taking a novel approach to the area of creating more treatment options for patients with cancer. The basic summary is that we're developing a rapidly adaptable therapeutic platform to treat patients with cancer using two platforms, one called PeptiCrad and one called PeptiEnv. The essence of the technology is that we use oncolytic viruses, which are viruses that are genetically engineered to selectively infect and kill cancer cells, and we coat them with tumor-specific peptides, creating essentially, using the oncolytic virus essentially as a carrier for the peptides, the vaccination strategy. We're spun out of the University of Helsinki, and we have raised just shy of 10 million euros to date, and half of that is non-dilutive funding from the Finnish government. We're just starting the Series A raise of 10 million euros, where we plan to use 5 million euros to fund the company through its phase 1 clinical program, and the other 5 million euros to fund the expansion of the platform, including PeptiEnv. The essence of what we're doing, you've heard from the previous speaker, is that with the immuno-oncology space dominated by the checkpoint inhibitors, the vast majority of patients don't respond, and those that do are the ones where there's a pre-existing immune response. So the question is, how can we take more of what we call the cold tumors and turn them hot? Can we use an oncolytic virus strategy to invoke a potent inflammatory response locally within the tumor to attract the immune cells that are necessary for immune stimulation, in particular CD8 positive T cell stimulation, and that could then drive a tumor-specific response. When people use oncolytic viruses, most of the immune response, of course, is to the virus, particularly with, for example, adenovirus, where we've all been exposed to adenovirus, so much of the immune response will be antiviral. And we're trying to, again, skew it towards the anti-tumor by coating the virus with tumor-specific peptides. So with this in mind, we have a very experienced team. Myself and Scott Cuthill. Scott's got deep experience in biopharma, 20 years experience in biopharma. Scott and myself are based in Oxford. I myself have a background as an MD, PhD in immunology. And we have a deeply experienced team in Finland, led by Sari Pessinen. The combined team has over 45 years of experience in the oncolytic virus space and the immuno-oncology space, having led one of the first companies in the oncolytic virus space called Oncos, now part of Targovax. So the team has worked together for the last 10 years and is familiar with the issues related to the fact that most tumors, if you look at the panel here, this is from published data from the Oncos days, where these are the tumors prior to intratumoral injection of oncolytic virus. And here you see the post-injection where you get a dramatic influx. The dark staining is the influx of the immune cells. But the issue is that, so that's really turning these tumors from cold to hot. But the issue is almost all of this activity is against the virus. So this is the human data based on this patient cohort, where you'll see 100% of the patients, of course, not surprisingly, exhibit an antiviral immune response, where only a few demonstrate an anti-tumor effect. What we're trying to do here is really skew the response to be tumor-targeted. This is from preclinical data in mice, where we've got the immune response happening in 100% of the mice and antivirus effect in a few of the mice. So that's the premise of what we're doing. How do we do that? We have technology that allows us to take advantage of the capsid characteristics of the adenovirus, which is net negatively charged over here. And we can take any peptide, add a polylysine tail, which is a net positive charge, to form an electrostatic complex, mixing the adenovirus with the polylysine tagged peptides, any peptides, forms a stable complex which survives freezing and thawing. Now, given that it takes simply 2 to 3 weeks to GMP manufacture a peptide as opposed to a virus, once you have a stock of virus, you can target any cancer that expresses any particular peptide target. That's the premise, if you like, of PeptiCrad. PeptiCrad standing for peptide-coated conditionally replicating adenovirus. Bit of a mouthful, but there it is. We have the same concept applied to envelope viruses, hence PeptiEnv, where we put a membrane anchor but have the same concept. We use adenovirus because we know that from the literature and the team's own experience that they really drive a CD8 positive T cell response and are almost certainly a superior adjuvant as opposed to the envelope viruses. We think that there's lots of reasons to believe it'll be synergistic with checkpoint inhibitors. This is our first lead product called PeptiCrad 1, which is essentially a adenovirus that has, like others, been engineered to selectively infect and kill cancer cells, but with the addition of 2 immunostimulatory molecules called CD40 ligand and OX40 ligand to really drive the stimulation of both arms of the immune system, driving a CD8 positive T cell response. And on top of that, we add, of course, we coat it with the tumor-specific peptides. In this context, and I'll come back to that when I show the pipeline, we're using NY-ESO1 and MAGE-A3, which are 2 tumor-associated antigens, which we know are well expressed on a selection of cancer types, which I'll come to. So in terms of the data, the preclinical data, we're a preclinical company. This just shows that by coating the virus with the peptides, it doesn't affect the oncolytic potential of the virus. And here's the data to show in a mouse melanoma model, a well-established B16 over model, the fact that this is tumor volume on the y-axis, days post-therapy and post-implantation of the tumor model. We prime 3 times, then boost 2 days apart. And this is the control showing very poor or very rapid tumor growth per control. This is with an uncoated oncolytic virus, the same backbone as PeptiCrad, which is the coated virus. And these are the individual animals where you can see the 5 animals with PeptiCrad showing very good tumor control. What about mechanism of action? This slide is again mouse melanoma looking at CD8 positive T cell responses. And the key is to look at the tumor specific. These are TRP2 and GP100, which are the targets on this particular model, showing the controls where the CD8 T cell responses in response to these specific antigens is no more than the background of the control, the negative control. The same happens with the uncoated, no peptide coating virus, but you're seeing an antiviral effect, of course, compared to when you actually coat, you're clearly getting a dramatic increase in the CD8 positive T cell counts. If you then combine that with checkpoint inhibitors, these are 2 separate models. One is a low MHC1 expression where you would not expect to see a very dramatic effect from checkpoint inhibitors or our own PeptiCrad. And indeed, again, this is tumor volume, days since tumor engraftment. These are the controls, rapid tumor growth. That's checkpoint inhibitor, the blue dot. The orange dot is PeptiCrad, but the combination shows dramatic tumor control. This is in a high MHC expression where you would expect to see a decent checkpoint inhibitor effect, which you do see relative to control. And again, with our own virus, uncoated virus, but then the combination. And these are the survival curves following and ongoing from these experiments where the controls are, of the 8 animals, 7 are now dead, and we haven't yet lost a mouse, and we're now re-challenging the combination group with the tumor to see if we have a memory CD8 T cell response and control of tumors. So this gives us a great deal of encouragement that the principle of a checkpoint inhibitor taking the brake off the immune system non-specifically in combination with pepticrad, which is really driving the CD8 positive immune response, we think is a very effective way to go. In the interest of time, I'm going to skip this slide. This just shows basically in a humanized mouse model a superior, vastly superior CD8 positive T cell response using our clinical peptide. This is the pipeline. Our first to go will be pepticrad 1 in combination with checkpoint inhibitor targeting melanoma, triple negative breast cancer, and non-small cell lung cancer. The reason for the choice of that basket is that these tumors, the majority of the cancers express those specific or tumor-associated antigens called NY-ESL1 and MAGE-A3. This is the clinical trial plan, and what you'll note here is quite novel in that we're able to take treatment-naive patients because we're able to delay the standard of care checkpoint inhibitor treatment by 2 weeks, allowing us to do the prime boost with pepticrad followed by the checkpoint inhibitor, which essentially the pepticrad is driving the immune response, the CD8, the tumor-specific immune response, and then we come along with the checkpoint inhibitor to take the brake off to really drive the effect of the combination. We've had our first meeting with the regulator, a scientific advice meeting where this plan was very well received. That was last week in Finland. So overall, then in summary, we have pepticrad and PeptiEnd as a platform approach to target any cancer, and in an era where we're looking at the neoantigen space, where there are many companies that are trying to find personalized, in other words, patient-specific neoantigens, we're able to use our technology to create pepticrads within 2 to 3 weeks, because we're able to manufacture a any polylysine-tagged peptide, and then mix it with our stock of virus to then inject into the patient. And what we expect is to be able to go from needle, biopsy of the tumor, to therapy within 4 weeks. And so we think this approach is going to be relatively non-toxic. Obviously, we haven't yet done the phase 1, and that's what we hope to show in the phase 1 clinical trial starting in January next year. And we hope to show that we get a superior CD8 positive T cell response and much more effective clinical responses than we're seeing with other treatments to date. So with that, I thank you very much, and leave you to your drinks for the late afternoon. Thank you.